everyone. I hope that you've all been enjoying the HOT conference today. It was really wonderful getting to see all your presentations, and we want to recognize the hard work that you've put in to attend this conference. I hope that you've been learning a lot about the exciting research happening right here in Texas among your fellow undergraduate colleagues, as well as gained additional insight for your future endeavors. So for the next portion of this event, it's my pleasure to introduce to you a pioneer of genomic research and biotechnology. Dr. Myers received his BS in biochemistry from the University of Alabama, his PhD in biochemistry from UC Berkeley, and completed his postdoctoral training at Harvard University. He joined the UC San Francisco Medical Center faculty in 1986 and continued his career as a professor and chair in the Department of Genetics at Stanford University in 1993. It was here at Stanford that Dr. Myers and his team participated in the public branch of the Human Genome Project, generating more than 10% of the sequencing data, including the entirety of chromosome 19. In 2008, Dr. Myers moved to Huntsville, Alabama to serve as the president and scientific director since the opening of Hudson Alpha Institute for Biotechnology. Beyond his research accolades, to me, Dr. Myers is also a close research mentor. I did research in his lab as a part of the BioTrain Academy at Hudson Alpha, and right away I was just blown away by the excitement and passion for discovery by Dr. Myers and his team at this institution. On the Hudson Alpha campus, Dr. Myers works closely with companies, educational programs like BioTrain Academy, and the Smith Family Genomics Clinic to streamline applications for human health and well-being. Dr. Myers' current research work aims to study the human genome with the goal of understanding how changes in the genome contribute to human traits, including human diseases and behaviors. So through the duration of this presentation, if you have any questions, feel free to just put it up in the chat or private chat it to me. But all things being aside, please join me in welcoming Dr. Myers for his presentation for the HOT conference.
Okay, thank you, Joelle. Hello, everyone. I understand that you've been going strong all day. It's sounds like it's been a very busy day. So I'm going to tell you about research that we do both at Hudson Alpha Institute, and then more of an emphasis on what we do in my laboratory. Now I'm going to try to share my screen. We'll see how that goes. Let's see. All right. So can you see that? I just want to be sure you see something with a title page that says Heart of Texas in the middle. Okay, good. And I need to move you all out of the way so I can see my slides myself, even though I know them. I'm not sure how you do this. I should know. This is probably my 3,000th Zoom I've had in the past year, and even some before. So we'll see. One thing that Joelle mentioned that she did work in my lab. I don't remember which summer. It was maybe 3 summers ago. As a part of our undergraduate research, one of our undergraduate research programs, I should say. There are multiple ones, and some of the work I'm going to talk about, she will recognize. So this is an aerial view of our campus. It's actually distorted. These 4 buildings. There's a 5th building coming up now. These 4 buildings. Look like they're taking up half of the space, but in fact, there's room for about 10 more buildings, 8 or 10 more buildings. Just by the way, Huntsville is where NASA started, even though there's a big NASA in Houston, obviously. And if you look up in the upper left-hand corner, that's Blue Horizon, the Jeff Bezos space, private space company. The building's complete. It's really, I love the space program, so it's really fun to watch that going on. So we do a lot of different things at Hudson Alpha having to do with the life sciences, a very, very heavy emphasis on genetics and genomics. So we're a nonprofit research and teaching institute. So we do research just like in a university. And we did a lot of genomics throughout my career before I came here from California. And we brought a lot of that with us and have, this is a field that moves really quickly, and we've been involved in both applying it and helping to develop some of those technologies. Okay, now we also have this unusual arrangement for a nonprofit to have where we have biotech companies that we that we're the landlord for them, and but they're in the same building, the same campus, and we have 47 biotech companies. They make up a huge portion of the 1,000+ people on campus, and they work on various life sciences things. We collaborate with them a lot as in the nonprofit side. I'm not going to show you or show you slides for this educational outreach part, but I encourage you. I'm going to show you the URL for our whole institute, but the URL for the education outreach program. I encourage you to do this. That's where the BioTrain program that Joelle worked in comes out of that. But there are 40 other programs that are part of that. It's a massive program. It's the largest in the world, actually, where you're doing outreach, outreach means a lot of things, but of course, we train the next generation of scientists in our labs and in the companies, but this is also education for teachers, for judges, for young people younger than college, certainly even very young, and a lot of different formats for that, and a whole bunch of stuff online that's free. And let's see, I can't see whether I push the advance for, let's see, let me push the advance so that you can see the URL. That's the URL for the education part, but just look through our whole page. There's a lot of stuff about COVID. We adapted very quickly to doing a lot of research, a lot of testing, antibody development of antibody treatments, wastewater testing, sequencing viral genomes to look for variants, sequencing people's genomes to look for genes that might be involved in making it better or worse for people. And so one reason I'm bringing that up is that on the education page, you will see something like 60 or more short videos that Dr. Neil Lamb, one of our faculty who runs the education outreach program, one of our faculty members, has done these short videos on various aspects of COVID that are extremely useful. I encourage you, just go look at one of them and you'll see what I mean. They've been viewed by 150,000 people. They really are everywhere. All right, so what do we do there? This is our faculty, our primary faculty. We have a bunch of joint faculty as well. And you can see from these Wordle things here that we do a whole lot of stuff. Much of it ties in with genomics technologies, which means ways of looking at DNA and RNA and looking at the sequences of DNA and RNA. You'll have to pardon me for just a minute. I'm going to try to get rid of the thumbnail. There we go. And and when we talk about disease, we work on essentially almost every organ system in the human body, a lot of different things. I'm going to show you a few of those. But we also notice this green agriculture. A significant portion of our work has to do with using genomics and genetics technologies to, we call it our plant genomics and sustainable agriculture program. And it really is, I'm not going to talk much about it, but it really is a remarkable group of faculty and scientists, students and postdocs and other scientists, computer scientists, working on improving crops, improving forests, and really trying to figure out how to do this and keep the planet sustainable so that we're not doing harm with agriculture, which is what happens with a lot. So I was, as Joel mentioned, I was involved in the Human Genome Project. Our lab first at UCSF, but then at Stanford, joined this public effort. There were thousands of scientists, and the idea was to figure out the sequence of the one human genome, one person's genome. Turned out it was a composite person, but just one genome. What the sequence means is figuring out the order of the letters in DNA, A, G, C, and T, because that order is really important, of course. And so you change the order or even change a tiny little portion, like one of those 3 billion letters in DNA, you can have a devastating or fatal disease. So we wanted to figure out how to get a reference human genome. My lab at UCSF, then we moved to Stanford early on, spent 13 years on this. A lot of scientists from around the world, really more than 2,000, and a lot of money. Much of our lab was, along with something called the Joint Genome Institute, did about 11% of this. And that was really hard back then because we didn't really know how to sequence DNA very well. And the technology had to improve along the way. But since then, it has improved more and more and more. So now you can sequence a human genome. We do a lot of this at Hudson Alpha. We've done more than 100,000 people's genomes, probably a lot more than that by now, many plant genomes, et cetera. And it takes a couple scientists, a lot of automation, just a couple days now, and about $1,000. And actually more cost goes into figuring out what to do with the data, interpreting it, storing the data, and sending it around, because it's a massive amount of data, than even generating the data. But it's very important to generate high quality data, and that's one of the things that we specialize in. So our groups, we have two big genome centers within our institute. One of them is the one that focuses on plants. That's the one on the left. And then a partnership that we created called Hudson Alpha Discovery that does all the rest of the sequencing. And when we talk about sequencing, we often are talking about sequencing the whole genome, but there's as much, if not more, work on sequencing portions of the genomes, panels of genes that are involved in cancer or brain disease or pharmacogenetics, or even looking at RNA, the expression of genes. And so there are many, many things that are based on using this technology for sequencing. One of our applications is actually, we have a small clinic. We're not a medical school, where Huntsville is a small city, about 500,000 people around the area, but doesn't have a big medical school. It has a lot of clinical work going on. We collaborate up with people all over, but we also have patients come in, a lot of times kids, but as many, even more adults come in, especially when they have an unexplained disease. I'm going to tell you something about that in a minute. So my lab, my interest has been for many years, several decades, in neurological disorders. And that really means neurodegenerative diseases like Alzheimer's, Huntington disease, ALS, et cetera, but also psychiatric disorders, which are much harder to figure out than the neurodegenerative ones. So that's what the rest of the talk is going to be. I'm going to tell you about how some of the stories there and how we've applied it. So here's one. This is old. Actually, we did this at Stanford. My graduate student, Len Pinocchio, was the one who essentially, we were studying a type of epilepsy that's in children. It's pretty bad news disease. They have seizures and ataxia. They're wheelchair bound, starting at about age 8 or 9. And it's inherited. It was due to a single gene. And so back then, this was in 1996, a long, long time ago, but it's a great example. Back then, finding a gene was a major effort, took lots and lots of times, lots of effort, lots of people. But Len found it by doing something called genetic linkage. And it turns out that it's really important. They found this gene, it encodes a protein that is an inhibitor of proteases. And it turns out if you give kids with this particular form of epilepsy, if you give them Dilantin, which is the drug that's usually used to prevent seizures, it actually is extremely harmful to them. It can even kill them. So you can use another drug that does what works a whole lot better. The reason this was important is when we found the gene, you could then say whether the child had this form of inherited epilepsy or another form. And the neurologists, while they're very good, could not tell the difference by looking at the clinical symptoms. So here you have a genetic test that's 25 years old now, where you can avoid treating someone with a common drug that most neurologists use and treat them with another drug. And it's at least does not harm them and helps them a lot. So switching now to more modern times, once DNA sequencing became less expensive, higher throughput, more accurate, we've been applying this now for years, and the technology gets better and better. I have a typo there. We've sequenced, this is a project, the beginnings of a project led by this fellow here, Greg Cooper, but a bunch of us participate in this, and we've sequenced 2,000 kids who have what used to be called mental retardation, but developmental DDID, developmental or intellectual disability. And we've actually done this for more than 2,000 children now. And we often sequence both parents' genomes too. And what's remarkable is that by applying the technology and really knowing how to interpret the data, and that's what Greg Cooper's team and a lot of people at Hudson Alpha do, are experts at doing. By interpret it, you can actually figure out the causes. About a third of the kids, 20, 30% now or so. And then another 10, 15%, we're pretty sure we know the answer and do additional work and can figure it out. So nothing works this well in biology. It's because we're sequencing early onset disorders and the technology has gotten so good. And we're choosing children who don't have what already known disorders like Down syndrome or Fragile X syndrome. We exclude, those are already figured out. These are other ones. And the reason this is important, this is about 3 to 4% of the world's population. So that's a lot of people worldwide are born with some disorder. And the vast majority, we haven't known the answers to why. And we can talk in a little later about why this is so valuable. Once we started then doing the same kind of sequencing and program to babies in the neonatal intensive care unit, now in 4 states, 5 states where we've actually expanded and with similar results. I'm going to just put that there. Please try to remember it. I'm not going to tell you now. It's a little teaser to say, why do we sequence both parents and the kids? If you don't know already, we'll talk about that. It's an interesting reason. So those are the places. And these are only 2 of our many programs for sequencing genomes of people to try to figure out what is wrong when they have a disorder. So now I'm going to shift to most of these are the ones I just talked about for developmental and intellectual disability are brain related, clearly. But now I'm going to talk about brain related disorders that affect adults as well. And this is on the left is ALS, then in the middle is Huntington disease. That's Woody Guthrie, Glenn Campbell with Alzheimer's disease, and Michael J. Fox with Parkinson's disease. So let me tell you how this works. So we did a study. Now this one's a few years old too, but it's continued since this. Then this was an interesting collaboration between our academic lab, nonprofit lab, David Goldstein's lab at Columbia University, and a pharmaceutical company, a small pharma company called Biogen in the Boston area. And so, and the reason we work with them is they study this and are trying to develop drugs for these disorders. ALS is this terrible movement disorder, fatal, affects people in their ranging between, sometimes can be quite young, but usually in their 30s, 40s, and 50s. The fellow up at the top is a famous New York Yankees baseball player from the 1930s, I believe, 30s and 40s, named Lou Gehrig, and so it's often called Lou Gehrig's disease. So by sequencing, we didn't even sequence the genomes then, because it was easier to sequence what are called the exomes, which just means the genes, the coding regions of the genes. You can select them out by hybridization and just sequence them. We don't do that anymore because it's better to sequence the whole genome and almost as inexpensive and fast. But we sequenced 3,000 DNA from 3,000 patients and a whole bunch of people who did not have the disease, these control individuals. You have to do that. You have to do the comparison. They need to be age and gender and ethnicity matched as much as possible. So that's what we do. And we identified 11 genes from that sequencing that had a serious impact on, they didn't necessarily cause by themselves, although some did, but they contributed to your burden of risk for getting ALS. And so 11 genes, this was new, and now there have been a few more that are found. 2 of these 11 are were great potential targets, and so they're being used by pharmaceutical companies to target new drugs for treatments, which is just wonderful to be able to contribute to that. Alzheimer's disease is much more common than any other neurodegenerative disease. It affects about 5.5 million people in the US. And as we're getting older and living longer, we're expecting another more than doubling of that in the next 5 to 10 years. It's pretty frightening, terrible disorder. Some people get early onset Alzheimer's. So we studied that. And I'm going to tell you why this is. Nick Cochran, who's a postdoc in my lab, collaborating with people in California and Colombia, South America. And basically, the short version of this is that there are families in Colombia, and it turns out this happens in other places as well, where they're actually people are related to each other probably 30 generations back, not realizing they are. These are from a founder mutation that was probably arrived from Spain and during the conquistador days when they came and took over the Americas and essentially planted a dominant gene, a mutation in a gene called presenilin-1, and all the people in this extended family have the same mutation in the same gene, which is really unusual. It turns out when you have that situation, this is fantastic for doing clinical trials because now you have at least the same genetic basis for the disease that you're studying and trying to get a drug to work. No drugs have worked for Alzheimer's disease yet, in spite of more than 30 different attempts at clinical trials. So we're hoping that this will make a big difference there. But there's an interesting part of this. There are some people, this is a single gene that when you have a mutation in this, you will get Alzheimer's disease. And usually early onset means in your 40s and 50s, maybe, as opposed to 70s and 80s. It turns out there's a small number of people in that family who have the mutation and they should get the disease. And they're so lucky because they don't, even when they're in their mid to late 70s. They haven't had it. So that's really, really unusual. So we have sequenced the genomes of these escapers, looking to see if we can find, is there another gene or a set of genes somewhere that protect against that mutation? And we're not there yet, but we're making some progress on this, and it's very exciting to maybe be able to find that. You know, if you think about this, if we knew a gene that if you had a particular variant in that gene, it helps to overcome a bad news mutation, that's a great target for drugs as well. So that's one of the reasons we do this, for that reason. I mean, for that purpose. So we've now, because that's been so successful, we've expanded this, and it's a big consortium throughout Latin America with several countries. And this is working closely with the neurologists and the scientists and the families throughout these countries. It's really a really exciting project. I love being able to do this because it's having a big impact and doing a whole bunch of things. And right now it's a couple thousand people with Alzheimer's and a couple thousand without, but this is expanding to be much, much bigger than that. Okay, let me look at the time. All right, I'm gonna switch gears, but you'll see how it's related, I hope, and start talking about gene regulation, how our genes are controlled, turning them on and off in the right cells at the right time and in response to the environment. And because I'm particularly interested, I'm interested in gene expression per se. It's what I studied in graduate school and still study as a basic science problem. But then we've been applying this to the human nervous system, those diseases I've been telling you about. So first, let me tell you about the ENCODE project. You may have heard of this or even read about it or done something with it. It's a big project. It's in its 16th year. It started right after the first human genome was sequenced. And the idea was we needed to have a big effort to try to figure out, what is all that 3 billion base pairs, what do they mean? What do they encode? What are their functions? So we've been able to be part of this very large project, probably 300, 400 scientists participating around the country and some other countries as well. It's an NIH funded, US NIH funded project. And we're basically doing a bunch of things, but one of the main things we do is to look at, if you see this line, ChIP-seq of hundreds of transcription factors, let me just explain what that means. We do an assay called ChIP-seq to see where proteins that are called transcription factors bind in the genome-wide, because those proteins, TF, you'll see me abbreviate it TF, but transcription factors control whether a gene comes on or stays off, meaning do you make a messenger RNA from it or not? And there are 1,600 or so of these transcription factors encoded by our genomes. So almost a tenth of our genome is tied into regulating our genes. So they're very, very important proteins. And so we study those a lot, and we study the regions of the genome where these proteins proteins bind and then regulate transcription. So don't get bogged down in the details of the technology. It's really fun, but I want to make sure that I get across to you why we're doing these and what the questions that we're trying to answer. So here's one. This is being led by a senior scientist in the lab named Jacob Loop with several other people in the group. And we work with post-mortem human brain tissue. This is after people have died, after people have died, pathologists, these are donors and families who donate their brains, and they're dissected into regions and then frozen, very cold frozen. We take those tissues, and one of the types of experiments we look within brain tissues is we try to figure out where a lot of these transcription factors are binding, because if we can figure out the basic place where they start to bind, that's the first step in whether a gene is made or not. And being able to control that step might be a way to treat disease, for instance, and I'll talk about that at the end. So Jacob has done this with a whole bunch of the tissues, many transcription factors. He also takes the cells after they've been frozen and does something called fluorescence activated cell sorting, FACS. He sorts the tissues according to the type of cell they are, and it's actually sorting the nuclei from the cells, because by freezing and thawing them, the cells break. The nuclei where the DNA is and a lot of the RNA is stay intact. And what you really don't worry too much about this, except those two circles we can do, or ovals, are two different cell types. The one on the left with the little red called nu-N-, those are glial cells, non-neuronal cells, and the ones on the right are neurons. So you can do that kind of separation and separate even more, so you can look at particular cell types. And just as an aside, you can look at even single cells within these kinds of studies and get really fine resolution about what you're looking for. And actually, I'm going to skip over this. It just tells you, but this is one example. So we take those nuclei and we do experiments, and we see where are transcription factors, particular factors bound, and where are regions within the genomes of each of those nuclei, where are regions of the genome opened up so that the gene can be turned on. That's called a TAC-seq, and the other one's called ChIP-seq. And the reason I'm showing you this is one of where you look through and you see where transcription factor binds and where open chromatin happens, and you see these little peaks are red. This is just one particular gene. We do this for all 21,000 genes throughout the entire genome, but here you're seeing with the peak, that means you have either open chromatin or transcription factor binding there. And this is information that you couldn't get any other way. So immediately we know about all the genes and where the factors are binding and what are they doing, because now we know this is a glia is a support cell for neurons, so it's a glial cell gene. Here's one that's neuronal specific, and you can see in the blue, you see places where this particular gene and this transcription factor are binding, transcription factors are binding. So those are in postmortem tissues, and we're limited in what we can do. So with those, so can we actually mimic this in the laboratory? So it turns out you can essentially culture human neurons. What you're really culturing is you're taking skin cells or cheek cells from somebody, and you can differentiate those. These are like stem cells, adult stem cells. You can differentiate them with different methods and turn them into, in this case, the upper are neuronal precursor cells called NPCs. Then you add something else to them and they turn into neurons. I'm sorry you can't see the bottom picture very well, but you actually get cell bodies with the axons and dendrites connecting, and even can get electrical signals passing in culture. So these are, and these are human, so they're kind of a mimic, a pretty good mimic for being able to do experiments to look at things like gene expression and many others in human neurons. So I'm not going to go over this other than to say that this picture, this data, and lots and lots of other data that we show that when we do work with the human neurons, it looks very similar to the postmortem brain tissues. It actually looks a lot better because it ends up being higher resolution. So we've learned an enormous amount from that, and some of that's going to follow here. So you might think, if you think about what I've talked about so far, we look for mutations, that spelling mistakes in genes that or maybe even cause a disease, or at least are involved in a disease, somewhere in this 3 billion base pairs, that we actually have 6 billion base pairs because we have 6 billion, 3 billion from mom and 3 billion from dad. Somewhere in there, there's a mutation, and we find some of those mutations that can cause a disease. So I'm also interested in gene expression. Are some of those mutations, and it turns out the answer is going to be yes, yes, yes. Some of those mutations are actually in regions or in genes that affect whether genes get turned on or off. So they're involved in gene expression. And so here's what an example that those experiments that I just told you about, and really all I want you to look at is this right here, this little line here. Are you seeing my arrow? I'm sorry, I don't know whether that shows up or not.
It does show up.
Okay, good. All right, so let's see. This line right here is part of the genome. And here we have where there's one particular sequence. You can see this peak here, all this gray. That means a transcription factor is this factor here is binding there. Then you see another donor. This is postmortem tissue where they actually have they don't have the same mutation or variant that this one had, and you see no binding. So it turns out 2% at least, and probably more than that, of all the binding events in the genome, and realize there are thousands of binding events, 2% of them bind to one version of the genome at a particular site and not the other version. So we call that allele specific or allele bias binding. And if you think about it, what if this variant, this DNA sequence variant, is when you get differential binding there, it almost surely affects a phenotype. So we're looking heavily in these regions of the genome to see if they're involved in disease. So this is just an example of one that it probably is involved in a particular disease that some genetics pointed to. And in fact, this slide's now 2 years old. I need to update it because the number is much higher than this now. But at least from a couple of years ago, at least 300 and something transcription factors. Remember I said we have about 1,600 that are coded. We know that, and we don't understand most of them yet. So probably half of the ones that have been studied, not just 20%, half of that have been studied are associated with a phenotype. And a phenotype usually means a disease. It can be things that are not as severe as disease as well, just traits. And so we're looking to see if these DNA sequence differences change when and where genes express and how much is expressed in it. And I'm not going to go through this one, it would take too long, but this is a great example of one that's involved in a disease that affects actually newborns. It's a really terrible disease called holoprosencephaly. Here's another example, and this was Drew Hartigan, was an MD PhD student in my lab. He's now doing his residency at Duke University. Drew we studied this. We had children who had developmental disability or developmental and intellectual disability, DDID. And we had a couple of children with those, or 8 children with mutations in a gene called EBF3. And by the time you get 7 or 8, you're pretty people with it, and it's a new gene that's never been seen before, you're pretty sure. But Drew did a bunch of experiments to show that mutations in this gene, and this gene encodes a transcription factor, that mutations in this gene change the ability of that transcription factor to bind to genes, to turn genes on. And so it was essentially molecular proof that this is what the cause. He showed that the mutant transcription factors did not bind and the non-mutant ones did. Okay. All right. I'm going to get to, this is I think this will be the last story I'll do so we can save a few minutes for talking. Now, this is something that's in progress. We're making a lot of progress. Other people in the world are making progress on these. There's some setbacks as well. But the idea here is for many neurodegenerative diseases are dominantly inherited. That means you've got a mutation in one of the copies of the gene and you get the disease. The dominant mutation in Huntington disease, Parkinson's, the ones we know, ALS, FTD, another type of dementia, Alzheimer's, the ones we know about, have a dominant mutation, and the dominant mutation doesn't knock the gene out. The gene, are we okay? The dominant gene is still expressed, and it makes a poison protein. That's why neurons die in these diseases. So we started a study a few years ago to try to figure out how do those genes get turned on and off. So we do a massive amount of work on just those genes, like the Huntington gene being one of the several of the Alzheimer's genes, et cetera. And we try to map all the places and proteins that turn them on or off. And then the idea is then to go to those places to see if we could turn the gene off by landing something there. And if we're not doing this clinically, we're doing it in those cultured cells I was telling you about, the cultured neurons. But the goal would then be to be able to deliver those repressors, probably through some kind of surgery. This has been done by Ionis Pharmaceuticals for a disease called spinal muscular atrophy 1. So the idea is to maybe turn those off. So that's what we're doing. Here's just a cartoon version. And on the left is the wild type version of the Huntington gene. It's making part of the protein there. The mutation of those darker colored circle, those are amino acids. And the mutant gene has a lot more of that particular amino acid. It's glutamine, it turns out. That's what the Huntington mutation is. So we want to turn off at the gene expression level, not at the protein level, so that this one is never made. And we've made a lot of progress in figuring out how to turn the gene on and off. Here's where we're turning it off in these neuronal cells. And we're working on doing that so that we can get to where we would develop maybe a compound that can do that. So it could even be delivered orally, maybe. All right, so I could tell you many more things, talk about many more things, but I'll stop there and let me see if I can open this back up where I can see you all. There we go. And happy to answer questions or do anything you'd like to now. So tell me what you're interested in.
All right, thank you so much, Dr. Myers. So interesting. All right, so we can open up to some questions. If you want to put something in the chat, or you can just say it out loud here. We can take a couple.
I realize that some of you are not biology majors, but you're science majors. So I hope I didn't overdo it on the technical stuff. I think figuring out, hearing what the goals are probably the more important thing there. So.
Yeah, I guess I can start if, as people are thinking of their questions, but what got you interested in this kind of genetics research specifically related to human health?
Well, I actually was trained in real basic science without any real applications in mind when I was an undergraduate and graduate school, and even part of my postdoc. But I started getting interested in human genetics because I was a biochemist. I was studying proteins and purifying proteins and studying how they do various things in vitro. And when I was a postdoc, I got interested in, I just heard about the fact that people were starting, and this was a long time ago, it was in the early 80s, that people were starting to map genes to figure out, even if they didn't have the genes yet, they would know that where on which chromosome a gene that causes a disease when it's mutated was located. And one of those first ones, not the very first ones, but an early one, was the Huntington disease gene. This is a gene we all have, and we make a normal protein from it. But when you have that mutation that I pointed out, you get the disease. And I just got fascinated by reading about that and decided late in my postdoc that I was going to kind of change fields. Although what I really did was I just added something to the field of gene expression and figured out how to meld them together. And so when we were part of the Human Genome Project, it had nothing to do with disease directly. We were sequencing a genome and part of this big team to do that. But I always had in mind that we would try to apply these to brain-related diseases. That's why that epilepsy project, for instance, happened kind of in the middle of the Human Genome Project, or even sort of in the earlier stages of it. So.
Okay, cool. Actually kind of related to the Human Genome Project, we have a couple questions relating to that. Kind of, what was the process of you getting involved and what was the experience like?
So I was at UCSF. I had been asked to write a they call them white papers. It's just kind of a summary paper about the technologies that people were using for various things. And I had been working on mutation detection methods, ways of detecting variants in DNA. And so they asked me to do this. And basically, it was that National Institutes of Health, the Department of Energy, the Sanger Center, they later became the Wellcome Trust in England, started proposing this idea. And well, actually, I had slides, but I removed them. I actually went to a meeting in 1984 because I was developing methods for mutation detection. I got invited, my boss, actually, my postdoc advisor, Tom Maniatis, got invited to go to this meeting in Alta, Utah. 19 people were coming. He didn't want to go. He wasn't working on human genetics. And because I was just starting to, he sent me instead. And it was 19 people. Alta is way up high in the mountains. It was in the winter, and half of us had altitude sickness the whole time. And basically, the goal of that meeting was a really interesting and important problem, both a social one and a scientific one, which is it was a collaboration between Japan and the United States in trying to figure out if when the offspring of the survivors now of the atomic bomb blasts in Hiroshima and Nagasaki, people survived it, then they had kids. Do their kids have more mutations than someone who was not exposed? And it turns out the technologies for measuring what we call the germline mutation rate, they just basically didn't exist. And so I'm at this meeting, I'm showing my mutation detection methods, and somebody and I was the only, George Church and I were the only youngsters there. I had no idea what they were talking about because I was new to the field. And somebody said the mutation rate is so low probably that we'd have to sequence the whole human genome to figure it out. And everybody laughed, and the Department of Energy, who supported that meeting, went back and said, this was like 1985, they said, we should sequence the human genome. That they were really the ones who came up with the idea of it. And I was just an innocent bystander. I really didn't know what much about it, but boy, did I get interested in it from that, because I got to observe it up close. So then the project started formally in 1990, but we were building the technologies and things for it. And my lab and a friend I was collaborating with named David Cox were doing mapping of genes. So that's why we became part of it. It was an amazing process to go from really having such crude technologies where it would have taken us 1,000 years to sequence the genome with the methods we had then, but they just got better and better and better, so that we finished it actually kind of early in 13 years rather than the 15 originally. And remember what I said, we can sequence a genome actually is in less than 2 days now. It's the technologies are just so much better. Joelle, I can see the questions. Do you want me to just go down them? Do we have time?
Oh, we can. I think we are kind of out of time, though. Shibni, do you think that we have time for one more question, or do we need to move on?
Yeah, let's do at least one more.
Okay.
Let's try. I can combine a couple. I think that.
All right, you do it.
There are some direct messages to me. So one question that was kind of asked by multiple people is, how do like the kind of genetic research that you do, how is it influencing or in relationship to like psychiatric disorders such as addiction disorders or like autism?
So well, autism is not really considered psychiatric. It is clearly neuro. We know the causes of some children with autism, but we're still trying to find others. And a lot of that research, I'd see somebody said, what kind of research do you do? It's actually, a lot of it is sequencing genomes of lots and lots and lots of people to try to figure it out. And a lot of advances have been made there. Psychiatric are things like bipolar disorder, schizophrenia, major depression, psychotic depression, various things like that. And I've been working on these for 20 years. It's really, really hard because the phenotypes, the actual diseases are very complex to describe. When you say you have Alzheimer's disease or Huntington's disease is a very clear example, there is very clear pathology. It can be imaged and it's due to a single gene. It's not true for psychiatric disorders. It's not true for many kinds of disorders. So we've done a lot of searching for genes. We have some that clearly are involved in it. but none of them are single genes like Huntington or cystic fibrosis. So that's it's unfortunate because these are really important disorders. They affect lots and lots of people worldwide. But nevertheless, we're also using these functional genomics things. We're actually screening drugs that are used for psychiatric disorders to see if we can find out if somebody's going to work better. Somebody will work that. This drug will work better in someone with schizophrenia, let's say, versus this person. That's been one of the places where some of the advances are made. We would like to see a lot more, and of course we. And when I say we, I mean the world is working pretty seriously on all this. So.
All right, cool. One more question. Can we do it?
Let's do it, Joelle.
All right, one last question. I think we got to move on. Okay, so finally it's, so you looked at kind of how certain genes can be turned on and off for coming up with some therapeutic ideas. Does your research ever also focus on maybe how to possibly change those genome sequences?
So that's gene therapy, and certainly I would have said gene therapy is a dirty word 10 years ago because it was not, it was so overblown and just very few advances in it. But because of CRISPR, the technology, the gene editing technology, and really a lot of other things that have advanced, it's getting better and better, and actually some of it is indeed working. Sometimes that goes in and tries to change the mutation, the actual fundamental cause. The real problem is how do you deliver it to the appropriate cells, even in a newborn, but certainly in an adult or a full-blown human being. Really hard if you need to get to particular cells. So some of the work is involved in trying to target the gene therapy to particular cells. So that's one of the things that we hope to see. I will say some of the same technologies could be used like that project I described for Huntington's and Alzheimer's, where we're trying to turn the gene off. That might be just as good, and it might be a lot easier. You just land something on the gene rather than trying to change the mutation. You land something on the gene that just shuts it off. And so people are working, and that involves genetics. Somebody wrote epigenetic phenomena too. So I think we're open. The world is open to both of these approaches, or all of these approaches, because I think we need them. They'll work in some cases and not in others, and so that's why you don't want to rely on a single approach.
All right. Well, thank you again so much, Dr. Myers.
Thank you, everyone.
It was so interesting to hear you speak.
I appreciate it. All right.
So final couple things is we want to take a picture of everyone in the conference attendees. And then right afterwards, the Hillis Scholars who are part of this event and Dr. Myers will click the link that I have sent beforehand. But thank you so much. All right, I'm going to remove the spotlight. And I guess, Shimini, did you want to handle the countdown, or how do you want to do this?
Yeah, okay. All right, everyone, if you want to be in the picture, please turn your camera on. We're just going to take a few pictures because there's a lot of us. So just smile and hold it. All right, ready? Caitlin, Nicole, whenever you're ready. 3, 2, 1, everyone smile. We done, Caitlin, Nicole? Yes, great. All right, Dr. Myers, thank you so much again.
All right, thanks everyone. Nice to see you all. Take care.
All right, the Hillis Scholars, you guys are welcome to go. Now we're going to move forward with the awards and wrap up the conference. All right, so I just want to quickly mention everyone who helped make this inaugural conference possible. It was definitely a joint effort from so many different organizations and people at Baylor, as well as other institutions who helped us pull this off. So thank you so much, especially to all the volunteers who were here with us all day. all of the speakers and all of the judges. We really, really appreciate your support of undergraduate research. So now we're going to go through and name all of the outstanding presentation award winners for each of the 17 divisions. So first up is chemistry. So in chemistry, Nam, you are the outstanding presenter, so congratulations. And presenters, don't worry about taking a picture or anything. We'll email you these awards after the conference. Okay, in bioinformatics and computational chemistry, we have Samuel.
You cut out.
Oh no, did you see the last award?
I think we saw Nom, and that was the first one.
Okay, I will go back. Sorry about that, Nam. Congratulations, you won in the chemistry division. So then in the bioinformatics and computational chemistry division, the winner is Samuel. In computer science, the winner is Ashley. In the biochemistry division, the winner is Thomas. In the biology 1 division, the winner is Faith. In the biology 2 division, the winner is Sanju. In the biology 3 division, the winners are Mary and Sai. In the biology 4 division, the winner is Caleb. In the biology cancer research and screening division is Arvind for his second presentation. In ecology and evolutionary biology, the winner is Soumya. In the environmental science division, the winner is Harrison. In the physics division, the winner is Kendall. In the public health and psychology 1 division, the winners are Brandon, Hannah, Madeline, Celine, and Jonathan. In the public health and psychology 2 division, the winner is Savannah. In the public health and psychology 3 division, the winners are Kara, Haley, and Lauren. In the public health and psychology 4 division, the winners are Catherine, Thunmei, Gabriel, and Ruth. In the neuroscience division, the winners are Kendall, Catherine, Laura, Riley, and Kayla. And that is our last division. Congratulations to all of the outstanding presenters. You all did amazing. To all of the presenters, I know from here, listening to the feedback from the judges, you all did very, very well. So congratulations. So with that, I want to thank each of you for being here today, taking time out of your Saturday to celebrate and present your research. You'll be hearing from us soon, so expect the feedback from the judges emailed to you. And then we're also going to be mailing you something special, so be on the lookout for that. And so with that, I also want to mention we're going to try to bring back this conference again next year. We're not sure what format that will be, but expect this to continue every year. So the second annual Heart of Texas Conference will be in spring 2022. So I hope to see you all there if you haven't graduated by then. But yeah, thank you all so much for being here. This concludes the conference.